So what followed after that was 5 more patients were treated in a similar way. And we found that we had beta amyloid reduction in 60% of them. So we could correlate the cavitation dose. So that cavitation is the effect of engaging the bubble, so having bubble activity during focused ultrasound. And the good news is that the beta amyloid reduction could be informed by the level of cavitation dose during the treatment.
Welcome to Curing with Sound, a podcast presented by the Focused Ultrasound Foundation. I'm your host, Allison Preston Smith. Dr. Elisa Konofagou is a professor of radiology and the Robert and Margaret Harari Professor of Biomedical Engineering at Columbia University. She directs the university's ultrasound and elasticity imaging laboratory, focusing on non-invasive, non-ionizing radiation and translational research using focused ultrasound for enhanced drug delivery, tumor ablation, neuromodulation, and more. An early pioneer in the field, Dr. Konofagou explores alternative imaging techniques for focused ultrasound procedures and investigates neurological and cancer applications in preclinical studies. She and her team also developed their own focused ultrasound devices to meet their research needs in clinical trials for Alzheimer's, DIPG, and more. Dr. Konofagou has won numerous awards and was the recipient of the Focused Ultrasound Foundation's Visionary Award in 2024. Welcome to the podcast, Elisa.
Thank you so much for having me.
How long have you been studying focused ultrasound, and what initially sparked your interest in this field?
I have been studying focused ultrasound for about 25 years now, and the initial interest was when I was a PhD student at the University of Houston and Texas Medical School. My PhD advisor was working on imaging with ultrasound. And then MD Anderson Cancer Hospital, which is a hospital in Houston and is very well known for cancer treatment, had interest in focused ultrasound and imaging of focused ultrasound lesions. So I had no idea what focused ultrasound was. And through that collaboration, we were able to image focused ultrasound lesions in liver using elastography, which was the imaging technique at the time that I was developing for my PhD for breast cancer.
Tell us more about your research using focused ultrasound in breast cancer treatment.
We're using focused ultrasound for ablation of primary tumors, and both benign and malignant. So we have a clinical trial actually that's on clinicaltrials.gov, and we are treating, but we also are informing the chemotherapeutic success through what we have a technology that uses focused ultrasound to mechanically vibrate the tumor. So this is more along the lines of imaging, but we want to see whether the response to chemotherapy changes the stiffness of the tumor. And the way we do it is that we literally knock on the tumor internally with a focused ultrasound beam and then evaluate the stiffness or the elasticity, because that's a biomarker of response. So the softer the tumor, the higher the response to the chemotherapy. So we want to inform right now the patients that their chemotherapy works instead of waiting multiple cycles, to inform them that it works within the first couple of weeks. And we have some very nice initial data to show that.
Oh, that's amazing. So could that essentially cut down the treatment time and the intensity of treatment?
Yes, that's exactly what we want to do. We want to cut down the time. So exactly what you said, if you already have a very soft tumor, or sometimes a tumor that has already completely disappeared, you potentially don't need the additional cycles that are very, as we know, very detrimental through the side effects, especially women undergoing breast cancer. But the other aspect is basically if the treatment doesn't work, and we can see that ahead of time, then they can be either an additional treatment or switch out the treatment to something else. So be able to get there ahead of time and be able to predict response is very, very powerful, I feel, for patients, especially in breast cancer, where it's a very difficult treatment for some women to go through without having success.
And you're one of the leading experts on blood-brain barrier opening and even developed your own device for conducting clinical trials. What makes your Focused Ultrasound device different from others?
So the most used right now focused ultrasound device is MRI guided. So that means that the focused ultrasound transducer has to be in an MRI scanner and use MRI to be able to guide the treatment, both for targeting but also monitoring of the treatment. Typically done with thermometry, MRI-based thermometry. So this is an excellent modality and has achieved a lot of widespread use in centers that can devote an MRI scanner for this for treatment. However, it does also constrain a technique that's typically low cost and real time to be confined in MRI scanners, and of course, with the associated cost to healthcare and also the associated time that's needed for treatment because MRI is not real time. So we wanted to build a system that instead of the patient going to the device, it's the device going to the patient, so we can wheel it anywhere the patient is. So and be able to treat them while either they're on the bedside or in the chemotherapy suite, and be able to use ultrasound imaging instead to perform real-time monitoring and be able to have a much faster treatment time.
What was the first clinical indication or the first clinical trial in which you had to develop your own device?
The first clinical indication that we went after was Alzheimer's disease, and that's because at the time when I joined Columbia, more than 20 years ago now, as I said, I was working on cancer and treatment of cancer, breast cancer in particular. And but a neurologist here, Scott Small at Columbia, he basically had a small DARPA grant and was looking to be able to use focused ultrasound for Alzheimer's, and that's how we got started. Obviously, in preclinical studies in mice, and then after we got our first NIH grant, we went to non-human primates, and after we showed the actual safety and efficacy of this portable device, we finally got FDA approval to go into Alzheimer's patients and show both safety but also efficacy in reducing beta amyloids.
So this was the first time you'd used a focused ultrasound device to open the blood-brain barrier. And what were your findings from this study?
Our findings were much more exciting than we initially anticipated. The FDA was very kind to give us the approval, but they also required that we only do one sonication, one microbubble administration, and at very, very low pressures on the order of what's used diagnostically. So I had a bet with my postdoc at the time, Adonis Pouliopoulos, who's actually in London, faculty right now at King's College, and that we're not going to open the barrier, that the pressure required going through a human skull that's much thicker than what we had in non-human primates, of course, much more inhomogeneous, that we're not going to open. So the good news is that we did open very close to where we intended in the prefrontal cortex. And the other very good news is that we reduced the beta amyloid in that region, because it's something that we have seen that in mice that we actually induce an immune response. So what that means is that you do open the barrier, and typically the intuitive aspect is that you open the barrier for the drugs to go in at higher dose, and that's how we got started. But there was no drug in that trial. We wanted to just see whether it's safe to begin with without a drug, with just a microbubble engaged by the focused ultrasound at the blood vessels. And then we realized because of the triggering of the immune response, we were actually seeing on the PET scan that the beta amyloid was actually, in fact, reduced, which is one of the hallmark proteins of Alzheimer's in the brain.
That's incredible. Those are amazing results. And what followed after that?
So what followed after that was 5 more patients were treated in a similar way, and we found that we had beta amyloid reduction in 60% of them, and then we were able to see that the amyloid reduction was due to the level of cavitation dose. So we could correlate the cavitation dose. So that cavitation is the effect of engaging the bubble, so having bubble activity during focused ultrasound. And the good news is that the beta amyloid reduction could be informed by the level of cavitation dose during the treatment.
You've also led clinical trials for blood-brain barrier opening and drug delivery in pediatric patients with diffuse midline glioma, or DMG. So what's the status on that project?
Yes, this is a project that uses the initial idea of opening the barrier for delivery of drugs. And we have completed the study with a specific drug called panobinostat that was successful in going through the barrier and then in this case not necessarily achieving tumor control. These are patients that unfortunately by the time of diagnosis that these tumors that typically start from the pons, they unfortunately have spread to the entire brain. So it's very difficult to have tumor control. But we were able to deliver panobinostat safely in a very difficult region, which is the back of the head of very young patients, pediatric patients, typically between the age of 6 and 17. And be able to have multiple also locations of openings. So now we're continuing. We have FDA approval to continue with a generic drug. So panobinostat is a proprietary drug, and now with a generic drug called etoposide, that we can actually have a study, similar study of opening the barrier 3 times a week in another 10 subjects to be able to see whether that generic drug, which can be much more affordable to patients, can be delivered in the same way.
Well, that will be a very important study, and hopefully you have great results from that. When it comes to blood-brain barrier opening, you've also worked with Parkinson's patients using that to deliver gene therapy. So what results have you seen so far?
So there is a way to have AAV delivery directly administered to the brain, but when this is done, first of all, it incurs multiple hours of surgery. And the reason is that if you were to inject directly into the brain without letting the brain accommodate the injection or the catheters that are pulled in the brain, the level of delivery is very low. So what typically is done with surgical interventions for drug delivery to the brain is that they need about a couple of hours to have the cannula stabilize in the brain and then inject the vector. And then they do that over multiple hours. So this typically in non-human primates, it lasts for 8 to 10 hours. So it's something that the gene delivery world knows that it's very difficult to translate to the clinic. So in this case, that's number one problem. Number two problem is that even if you were to have AAVs delivered, it's very difficult to reach the neurons. So the target is the neuron, and in order to be able to reverse the degeneration. And unfortunately, even after several hours of having this surgical procedure in the brain for direct injection of AAVs is done, the main cells that do get transduced are typically astrocytes which are basically not directly involved in the degeneration. So it's very difficult to express this gene delivery vectors in the dopaminergic neurons. The dopaminergic neuron is a very important, obviously, neuron and the most affected in Parkinson's. So we were able to show both with non-invasive procedures. We know focused ultrasound is a non-invasive procedure, transcranial procedure. Without opening the scalp or the skull, we were able to deliver the AAVs to the dopaminergic neuron. So we're very excited about these results. And hopefully we can translate this one day to Parkinson's patients.
Mm-hmm. Are there any other indications that you're interested in researching?
Yes. So one of the main interests that radiation oncologists and neuro-oncologists have is brain metastasis to the brain. So over the past couple of years, we're working towards getting FDA approval for the non-small lung metastasis to the brain. And that's something that Focused Ultrasound Foundation has been helping us with.
And any other potential focused ultrasound devices that you and your team will be looking to develop?
So we're very excited about having a very portable, even more portable device than the one that I mentioned, and going to the point where you can have a laptop and an imaging laptop to be able to open the barrier. And I know it sounds a little bit far-fetched, but we are making the first step towards using an imaging scanner to be able to open the barrier. So this is, as I mentioned, very important for translational performance. And the fact that we are using very low pressure, as I mentioned earlier, diagnostic ultrasound level pressures, we can use a diagnostic ultrasound scanner to open the barrier. So that opens a lot of different applications and potentially also with existing resources in hospitals to be able to use this modality for treatment in the brain, but also elsewhere. So in this case, we have what we call theranostic ultrasound because we use it for both therapeutic and diagnosis. So, and at the same time, we can open the barrier, but also image what happens to the barrier with a diagnostic ultrasound scanner. And that diagnostic ultrasound scanners have been becoming more and more miniaturized. There's everything, from a laptop to an iPad to an iPhone and now wearables that can do that. So we're riding that technology wave, if you will, with that.
That would definitely be helpful. And it sounds like this is one of the barriers to this kind of technology. Are there any other barriers that you see with focused ultrasound?
I mean, the main thing is what the Focused Ultrasound Foundation is doing so well, and is basically putting focused ultrasound at the front, center of technologies that should be used more in the clinic. But with essential tremors, I think it has been a groundbreaking way of adopting a technology for the brain. And I think it's not on everybody's radar of what can happen with focused ultrasound going forward. But I feel like ablating the brain is only the beginning. And I think having the brain being modulated, so stimulated by the focused ultrasound and opening the barrier with focused ultrasound, like what we discussed before. And I forgot actually to say another application that we actually very much looking at clinical translation, which is modulation with focused ultrasound. So not opening the barrier, no macro bubbles, but just as ultrasound alone could have an effect in the brain, but also in the peripheral nerves for treatment of neuropathic pain. And we're looking at that with the help of the Focused Ultrasound Foundation to be able to stimulate nerves and to suppress pain sensation. And it looks like right now, especially with the carpal tunnel syndrome patients, we are seeing a lasting effect of a suppressed pain, significant suppressed pain by applying focused ultrasound at the periphery. And we hope to do that also with applications such as facial pain that's very devastating. And I'm finding out more about these conditions to patients. And with our neurosurgeons, here at Columbia, we should be able to translate this also to the brain for pain treatment.
So, Columbia University was recently named a Focused Ultrasound Foundation Center of Excellence. How do you plan to work more closely alongside the foundation with this new designation?
Yeah, so we're very, very honored, extremely honored to have this designation. We want to work with the foundation into having this technology that I mentioned more widespread in community hospitals or hospitals that not necessarily have even an MRI on their site, but they can wheel this technology or these systems to, as I said, to the bedside of patients, worldwide. We have an advantage here because we do have an amazing medical school and an excellent engineering school. And I have been, as a faculty here, having basically appointments in both schools. I have benefited, my research has benefited overall, but also now the Center of Excellence can also benefit. And we're talking about neurosurgery, radiology, neuro-oncology, immunology, oncology. So there's a lot of that aspect of all these multiple departments that are very collaborative. So this is another ecosystem at Columbia where you are able to collaborate with all these wonderful physicians and physician scientists in this case. And then the engineering aspect. So obviously the amazing biomedical engineering students that we have that are able to synthesize this knowledge and be able to crunch through all these different papers and different disciplines.
Where do you see the focused ultrasound field heading in the next 5 to 10 years?
That's a great question. I think cancer applications are not going away. We are making great strides, obviously, in cancer treatment, but there are a lot of cancer patients that we still lose to the disease. So I would see it as a technology that can be used to basically treat resistant cancer anywhere in the body, and then be able to have extend the survival of patients that are either having resistant primary tumors to standard treatment or metastasis to other organs, and then be able to have something, a technology like this one, to either open the barrier like we do, or ablating, either with mechanical ablation, such as histoscopy is doing, or thermal ablation, like we do for the breast cancer aspect. But also what we have been looking at more recently is how you can also induce with focused ultrasound what's called sonoporation. So this is very well-known effect, and a lot of people have actually been reporting this. But you can also use that on site to be able to increase the chemotherapeutic dose, similar to what we do in the brain tumors in the brain. But you can also do that, for example, in breast tumors, and we have found that this actually works very well in breast tumors as well, and triple negative breast tumors, which are very more resistant to therapy.
Well, thank you so much, Dr. Konofagou. It has been so great talking to you and hearing about your important research and all the work that's going on in your lab.
Thank you so much for having me.
How can our listeners keep up with you?
You can go to url.bme.columbia.edu to find our latest publications and our latest conference presentations. As I mentioned, we also are listed in clinicaltrials.gov for a few of our studies that we have ongoing. And I think we're active on Twitter and then also on LinkedIn.
This episode of Curing with Sound has been presented by the Focused Ultrasound Foundation. If you'd like to learn more about Focused Ultrasound or the Foundation, visit our website at fusfoundation.org. You can also sign up for our newsletter and follow us on social media. Be sure to subscribe to the Curing with Sound podcast so you don't miss the latest information about Focused Ultrasound. You can also rate and review the show.